Presentation is entitled Genetic Purity of Two Commercially Cultivated Peanut Varieties in Ghana, and our presenter is Jennifer Abogoom.
Good day to everyone. My name is Jennifer Abogoom. I'm an MPhil student in Science and Technology at the Kwame Nkrumah University of Science and Technology in Ghana. And today I'll be making a presentation on my research topic, genetic purity of two commercially cultivated peanut varieties in Ghana. And in this presentation, I'll take you through an introduction, give you a little information on the materials and methods, present the results and discussion, as well as conclude and give recommendations based on the results obtained in this study. In recent times, Ghana has experienced an increase in the development and release of new peanut varieties in the country, thereby giving an opportunity for the country to increase its production. However, reports indicate that less than 5% of the peanut seeds used by farmers in the country is certified seeds sourced from the former system. This means that about 95% of the seeds that farmers use in the country is sourced from either from the market or from their own saved seeds. Now, it is important that the genetic purity of the varieties is maintained, especially for improved peanut varieties, so as to obtain better yields. This is because for the farmer to be able to enjoy the trade that have been incorporated in the improved varieties, there needs to be a maintenance of the genetic purity of that variety. Seed quality testing is an important aspect of crop production for farmers, especially. This is because farmers are better able to know the planting value of their seeds before they even plant. And the genetic purity of the seeds seeds used by farmers can be determined when seed quality is being done. However, in Ghana, the testing of the genetic purity of seeds used by farmers before planting is hardly done. This can be attributed to either lack of knowledge on the part of farmers on the various seed quality testing methods, or limited access to resources by farmers on seed quality testing methods. This means that the genetic purity of the peanut varieties that farmers use in the country are unknown. This study involves two peanut varieties that are commercially cultivated in the country, Chitauchi and Yenyawoso. Chitauchi, also known as Chinese, was introduced into the country over 40 years ago, and Yenyawoso was introduced in the country just in 2012. These two peanut varieties are highly preferred by farmers, mainly due to their early maturing trait, as well as their other qualities that they present. These two varieties possess the potential to increase the productivity of peanuts in the country. This is because reports indicate that about 90% of the land area used in peanut production is being cultivated by Chitauchi alone. And Yenyawoso is a variety that has improved traits in resistance to rust. And also the kernels of the seed contain about 50% oil, which makes it suitable for commercial oil extraction. The results of the study is important because the study of this result will help relevant stakeholders in the country to make informed decisions, policies, and measures that will ensure that farmers in the country use peanut seeds that have high genetic purity. Therefore, the study sought to assess the genetic purity of farmer-saved seeds of these two peanut varieties. And in doing so, we chose to use molecular markers to assess the genetic purity of these peanut varieties, owing to the fact that molecular markers have a high advantage over the conventional methods of assessing genetic purity. To conduct this study, we obtained a total of 28 farmer-saved seed lots. And out of these 28, about 20 of these seed lots belong to the Chitauchi group, and 8 of these seed samples belong to the Yenyawoso. To serve as a check or reference in this study, we obtained seed lots from the two main research institutes in the country, which is the Crop Research Institute and the Savannah Agricultural Research Institute. And one Chitauchi seed lot was obtained from the Savannah Agricultural Research Institute, whereas 2 Yenyawoso seed samples were obtained from both research institutes to serve as our check in this study. In order to obtain genomic DNA from these varied plants, we randomly selected 15 seeds from each seed lot and then planted them in a plastic container in the plant house. Subsequently, DNA extraction was performed on just 10 individual plants from each seed lot, representing a particular seed lot. The quality and quantity of the genomic DNA was determined using a nanodrop spectrophotometer, and subsequently, the concentration was diluted to about 100 nanograms per microliter. And further, equal amounts of the DNA from the 10 individual plants representing one seed lot was taken to form a bulk for that seed lot. And these bulk DNA samples were then used for the PCR analysis. 25 SSR primer pairs, which were randomly selected from already existing literature, were used in this study. And what we did was that we initially screened these 25 SSR primer pairs against our 3 checks to select those primer pairs that are polymorphic for these 2 peanut varieties. And out of the 25, only 4 primers were polymorphic, and these 4 were subsequently used in assessing the genetic purity of the 28 farmer-saved seed lots. After amplification, the products were separated in a 6% polyacrylamide gel, and these gels were then run in a vertical electrophoresis unit, and an alpha imager was used to visualize the gels, as well as to take a picture of those gels. Each band in the gel was considered an allele, and so each allele was scored, and the method we used the presence and absence of each allele that we saw in the gel, and then we scored that manually. Then a similarity data matrix was obtained using the simple matching coefficient, and the similarity coefficient was then used to construct a dendrogram following the UPGMA and the SAHN method. All the data analysis was done using the NTSYS-pc software. Results and discussion. Figure 1 is an image of the gel for primer SEK17F6 that shows the genetic variation among the 31 granule seed lots. The first well loaded was the ladder, and CH represents the Shitauchi seed lot that we obtained from SARI. YS and YC represents the Yenyawoso seed lot as the check that we obtained from both institutes. And wells 1 to wells 20 represents the farmer-saved seed lots that were grouped under the Shitauchi variety. And wells 21 to well 28 represents the Yenyawoso seed lots that belong to the farmers. This figure is a dendrogram that shows the genetic relationship among the 31 peanut seed lots that were generated using the UPGMA and the simple matching coefficient. Basically, the cluster analysis grouped the 31 peanut seed lots into 2 main groups, and further, it grouped them, which we have cluster 1 and cluster 2. And in cluster 1 contains basically seed lots that belong to the Yenyawoso variety, and cluster 2 contains seed lots that belong to the Shitauchi or the Chinese variety. Now, it's to be observed that in cluster 1, only 2 of the farmer-saved seed lots were clustered at 100% similarity index with our 2 reference Yenyawoso seed lot from the 2 institutes. And in cluster 2, only 5 out of the 20 seed lots that belong to the Chinese group were clustered at 100% similarity with the Chinese or the Shitauchi peanut variety that we obtained. from the Research Institute. This means that these varieties, these seed lots that belong to the farmers are 100% genetically similar to our reference seed lots from the Research Institute. And away from that, we have the other farmer-safe seed lots clustered in the various groups, sharing various amounts of similarity with our reference checks. This may be because this means that the genetic purity of those farmer-safe seed lots have reduced, and this may be due to the different artificial selection techniques that various farmers use. And then also because farmers in the country mostly do not replace their seeds after the first purchase, and they use seeds that have been saved for more than 2 to 3 years in their production. And naturally, this would lead to a reduction in the genetic purity of those seeds. Interestingly, one seed lot, which is cluster 1, that is UEF2, was given to us by farmers as a Chinese or Shitauchi seed lot, but in the cluster analysis, this has been clustered in the Yenyawoso seed lot. Also, a farmer-safe seed lot, which is UWF1, which was given to us as a Yenyawoso seed lot by the farmers, was clustered in cluster 2 along with the seed lots that belongs to the Shitauchi or the Chinese. This means that these two seed lots have been placed in the wrong groups, and this may be as a result of APRES checks from mechanical mixtures of during farmers' fields or during processing or storage. As I said earlier, out of the 20 farmer-safe Shitauchi seed lots that were used, only 5 of them were 100% genetically similar to the reference Shitauchi seed lots. And out of the 8 farmer-safe seed lots that belong to the Yenyawoso that were used in this study, only 2 of them were 100% similar to our reference Yenyawoso seed lots obtained from the Research Institute. Now, 2 of the farmer-safe seed lots, which is the UEF2 and UEF1, were grouped in clusters other than the clusters that were expected to be found in. In conclusion, the genetic purity of Yenyawoso and Shitauchi varieties, per the results that were obtained in this study, shows that they have declined in the hands of farmers when compared to those that we obtained from the Research Institutes. And we are recommending that smallholder farmers are trained on how to maintain genetic purity of their seeds after the first purchase, as well as they should be sensitized on the need to purchase certified seeds every 2 to 3 years after the first purchase. I would like to acknowledge the support of the BHEAD program, as well as the Peanut Innovation Lab, as well as my advisors, Professor Richard Akroma, Dr. Robert Edu, and Dr. David Jordan for their support in this study. Thank you very much for your attention.
Thank you, Jennifer. Very nice presentation. We have time for questions. Do we have questions from the audience? And while we're waiting for any questions, I have a question for you, in that as you're seeing the purity of those 2 varieties change in the hands of the growers, is there any, what's the most common change that's happening? Are there some common varieties, older varieties, that are replacing them or being ingressed, or is it just a mix of new introductions or new materials that are coming into and changing the diversity within the lines? I guess I'm asking is, what's most likely to affect the purity? Is there certain varieties from the country that they would be familiar with?
If I get your question right, the question is what is most likely to change the genetic purity of those varieties, right?
Yes. Are there certain popular varieties that the farmers use that are replacing them, or is it just a lot of different varieties which are changing the purity of the ones that you want?
As I said earlier, the Shitauchi is a variety that has been introduced in the country for over 40 years, but farmers in the country still grow this variety due to the early maturing nature of that variety. And what's mostly is causing the reduction in the decline in the genetic purity of that variety is because after its release, farmers usually don't go back to purchase those variety the seeds from the certified sources. And so, and they as well plant in their lands, plant other varieties near those varieties. And in the process of harvesting and handling, post-harvest handling, they do not take care to really separate these processes. And so in the process of this, mechanical admixtures or mixtures happen. And so this might cause the reduction in the genetic purity of that variety.
Excellent. Well, thank you very much. Our time is up, and thank you for your presentation.
Thank you too.